This week, we're going to go through the second part of diseases caused by helminths. The lecture aims to identify and describe the following flatworms: beef and pork tapeworms, Taenia sanguinata and Taenia solium, fish tapeworm, Diphyllobotrium latum, the sheep liver fluke, Phaseola hepatica, and blood flukes, Schistosoma mansonis, Schistosoma japonicum, and Schistosoma hematobium. The aim of this lecture is also to identify and describe the flatworms on the previous slide based on their morphology, life cycle, distribution, associated human diseases, signs of infestation, and control and prevention. This lecture also aims to describe the following human diseases associated with flatworms: toniasis, neurocysticercosis, diphyllobotriasis, fasciolisis, schistosomiasis, and swimmer's itch. As well, this lecture aims to describe the human diseases associated with flatworms on the previous slide based on clinical symptoms, epidemiology and transmission routes, laboratory diagnosis, treatment, control, and prevention. Cestodes are elongated, segmented, hermaphroditic flatworms that inhabit the intestinal lumen. They have a head, neck, and tape-like body consisting of segments called proglottids. Larval forms are cystic or solid and inhabit extraintestinal tissues and organs. There are three types of tapeworms: the beef tapeworm, Taenia sanguinata, the pork tapeworm, Taenia solium, and the fish tapeworm, Diphyllobotrium latum. Infections with the beef tapeworm usually cause mild epigastric discomfort, nausea, flatulence, and diarrhea. However, cases can pass proglottids in their feces. Although humans are the definitive host, this parasite can also infect cattle, so eating raw or undercooked beef is the main route of transmission. This is part of the parasite life cycle, which will be described in detail on the next slide. The disease is rare in North America and only occurs on cattle feedlots where disposal of human sewage is inadequate. In these areas, cattle ingest ground vegetation contaminated with eggs from infected humans. Embryos hatch in cattle intestines, penetrate the intestinal wall, and migrate through circulatory or lymphatic system to the muscles. They transform into white, ovoid, cystic forms in tissue, producing a spotty or measly appearance. Humans ingest undercooked beef from infected cattle. The larval cysts, or cysticerci, develop into adult tapeworms, which can survive for many years attached to small intestines. They grow in length by increasing proglottid segments. The proglottids mature and produce eggs, which are shed into the environment with fecal matter. This is a picture of an adult Taenia sanguinata tapeworm. They're usually 4 to 6 meters long, which is around 15 feet long, if imperial measurements are used. Some may grow to as long as 25 meters or 80 feet in length. This is actually longer than 11 Shaquille O'Neals lying together from head to foot. The head is at the far right and is the smallest part of the worm. The head is also called the scolex. The beef tapeworm has 4 sucking discs and no hooks. You can see 2 of them clearly in the picture on the left. On the top right is a proglottid, which is a segment of the tapeworm which has been stained with dark Indian ink to show the uterine branches. On the lower right is the tapeworm egg. It is highly magnified as it is only 30 micrometers in diameter and is usually found in the uterine branches. Both beef and pork tapeworm eggs look alike and cannot be distinguished from each other. Beef tapeworm infections are diagnosed in the laboratory using microscopic identification of eggs or proglottids in fecal or stool specimens. These specimens should be collected on 3 different days. As mentioned on the previous slide, Taenia eggs cannot be differentiated, so the number of uterine branches in the proglottids is used for differentiation between the 2 main species of Taenia tapeworms. The beef tapeworm has many branches, 15 to 20 on each side, whereas the pork tapeworm, which we'll discuss next, has fewer branches, only 7 to 13. For lab-confirmed infections, treatment with anti-helminthics is available. The best way to prevent beef tapeworm infections is to cook meat to safe temperatures, at least 63 degrees Celsius, measured with a food thermometer placed in the thickest part of the whole cuts, and 3 minutes resting before serving, and at least 71 degrees Celsius for ground beef. Inspection of beef is also carried out by meat inspectors. Symptoms of pork tapeworm infections are similar to beef tapeworm infections. However, there is one big difference. Ingestion of pork tapeworm eggs or gravid proglodids result in cyst forms causing serious disease. This condition is called cysticercosis. Although there are generally no symptoms, lesions can develop in tissue and organs. Cysts in the brain and spinal cord can result in neurocysticercosis and seizures or epilepsy. Psychiatric and neurological abnormalities such as confusion and imbalance may also occur. Neurocysticercosis may also cause strokes and become life-threatening. Like beef tapeworms, the definitive host of pork tapeworms are humans. Therefore, eating undercooked pork results in tonisis caused by the pork tapeworm. The other disease associated with pork tapeworm is cysticercosis, which is already described on the previous slide. However, we need to go over the route of transmission again, since it can be confusing and does not occur in the beef tapeworm. Cysticercosis can be transmitted directly by eating eggs from individuals infected with the adult worm through the fecal oral route, or indirectly through food or water contaminated with eggs, or by autoinfection from poor hand hygiene. Pork tapeworm infections are rare in North America and occurs mainly in rural areas with poor sanitation, where free-range pigs roam freely and feed on human feces. To understand the routes of transmission a little better, let's look at the life cycle of the pork tapeworm. When human or pigs ingest eggs or proglodids, embryos hatch and penetrate the intestinal wall, migrating through the circulatory or lymphatic system to muscles, organs, and the brain. They transform into cyst forms in tissue and are called cysticerci. In humans, this condition is called cysticercosis, as indicated in the top of the diagram. When humans ingest undercooked pork containing cysticerci, they develop into adult tapeworms in the small intestines, and the condition is called tonisis, as indicated in the bottom of the diagram. Adult tapeworms may survive for decades. They can grow to 2 to 4 meters or 6 to 12 feet long. The cysts, or cysticerci, may remain viable for up to 5 years. They're 1 centimeter in diameter, white and opalescent, and can be seen without magnification. The image shows the presence of large numbers of cysticerci in pork muscle tissue. The picture on the left shows the head or scolex of the pork tapeworm. It has 4 sucking discs with a double row of hooks. The proglottid segments and chains are shorter than those in the beef tapeworm and only have 8 to 12 uterine branches, which is less than those present in beef tapeworm proglottids. These differences enable these two tapeworms to be differentiated, as the eggs are identical. Both have radial striations and hooks that can be seen within. Tonisis is diagnosed by the microscopic identification of eggs or proglottids in fecal specimens collected on 3 different days. Since eggs of Taenia tapeworms cannot be differentiated, the morphology of the proglottids are used to distinguish beef tapeworms from pork tapeworms. Pork tapeworms have lower numbers of uterine branches than beef tapeworms. Neurocysticercosis is diagnosed by brain scans using either computerized tomography (CT) or magnetic resonance imaging (MRI). Alternatively, serology or tissue biopsy can also be carried out for lab confirmation. Pork tapeworm infections can be treated with anti-helminthics. Depending on the location, number, and size of cysticerci, corticosteroids may need to be prescribed as well, because killed cysts may provoke inflammatory responses. Neurosurgical removal may also be required in the case of neurocysticercosis. Treatment of individuals with toniasis is required to prevent cysticercosis in close contacts, such as family members. Good hand hygiene to prevent fecal-oral transmission of cysticercosis is essential. To prevent toniasis, pork should be inspected and also cooked thoroughly before consumption. Now let's look at the fish tapeworm. Infections with the fish tapeworm, which is usually limited to one worm, is mostly asymptomatic, or there are mild symptoms such as abdominal discomfort, diarrhea, vomiting, and weight loss. Infection with a large number of worms may result in intestinal obstruction. Gallbladder disease may also occur. One of the more unique features of fish tapeworm infections is pernicious anemia, resulting from the worm's striking affinity for vitamin B12. This causes low red blood cell levels due to vitamin B12 deficiency. Fish tapeworms are acquired by eating raw or undercooked fish, usually from the northern hemisphere. The definitive hosts are humans and other fish-eating mammals. Fish tapeworm infections are found in temperate and subarctic climates of the United States, Canada, Europe, and Asia. They may also occur in other parts of the world where infected fish are transported to and consumed. There are two intermediate hosts in the life cycle of the fish tapeworm. Eggs from infected humans are passed in feces to contaminate fresh water. Ciliated larvae hatch from eggs and are ingested by freshwater crustaceans. The larvae develops in these copepores, which are the first intermediate host. The second intermediate host, usually a small fish, eats the copepod. Larvae are released to develop into a more advanced larval stage in fish muscle. When humans eat undercooked or raw infected fish, the larvae develop into a long tapeworm in the intestines. It is the largest tapeworm in humans in terms of its broad size and can reach 10 meters or 30 feet in length. There are two sucking grooves for attachment, and the proglottids are wider than their length, so it is also known as the broad tapeworm. The uterus are rosette-shaped, as can be seen in the top image. Eggs are smaller than 1/10 of a millimeter and need to be viewed using microscopy. Diagnosis is by the detection and identification of eggs or proglottids in fecal specimens. Anti-helminthics are available for treatment. Preventative measures include proper treatment or disposal of human sewage, prohibition of sale of fish from heavily infested areas, avoiding the consumption of raw or undercooked freshwater fish, including fish that migrate from seawater to spawn in freshwater, for example salmon. As well, adequate freezing and cooking of fish will kill the parasite, and this is a method that some well-trained chefs use in preparing dishes like sushi.